Okay, I guess we should go ahead and get started since I understand we are on a timetable. I don't think there's necessarily a hook, but I'm sure we'll be prodded to move things along if we run a little late. I'm Tom Ferkol, who I've just discovered 5 minutes ago that I'm older than I think I am, and this is Phil Bailey. We are two members of a team that has a project that's supported, as Alan alluded to, by the Children's Discovery Institute. And again, as Alan pointed out, I think it's really important, I think this is what truly makes us unique, is that we crossed an awful lot of disciplines to put this project together, including myself, who takes care of kids, with Phil, who takes care of mechanical engineering, and Susan, who takes care of genes, and Steve takes care of older people, like me. But we've all been very much interested in understanding how cilia work. And let me tell you a little bit about what cilia are. Now, the cilia are tiny little organelles, these tiny little hairs that have a myriad of functions in the human body. In fact, you can find them pretty much in every tissue in the body. If you look at sensory cilia, you can find them in the rods and cones in your eyes. Those are the cells that allow you to see. You can find them in your kidneys, in your biliary tract, in your gut, which helps your body know whether or not you got fluids moving through at a reasonable rate. Because I'm a lung doctor by training, we've been particularly interested in the respiratory cilia that line the airways, the eustachian tube that feed into the middle ear, and also the sinuses. And it also is the same structure that you see in a flagellum that causes the sperm to move. And if you have defects in cilia, they can result in a myriad of diseases. As you already kind of alluded to before, you have these sensory cilia here that can affect kidney cells, liver cells, pancreatic cells. They're in your bone, they're in your cartilage, they're in your retina, they're in your ear, they're in cells that are designed to repair your body. And if these are defective, they can result in a wide range of diseases. I mean, the classic example that's only been recently recognized within the past decade is that if your sensory cilia in the kidney doesn't work, you develop something called polycystic kidney disease, which is the most common reason for kidney transplantation in adults. If you have a nodal cilia, which is a cilia that only is found on the surface of the developing embryo, if that doesn't work, then you have left-right laterality defects. And so, your heart's on the right, your liver's on the left, and your spleen, God knows where it is. And so it results in kind of unusual abnormalities in the positioning of organs, and it also leads to congenital heart disease. And this was not recognized, again, up until maybe about a decade ago. Because, again, like I said, I'm a lung doctor, I'm particularly interested in what happens in the lung. And there is a motile cilia that beat, that literally sweep bacteria out of the airway or out of the middle ear or out of the sinuses, which keep those organs sterile. So to demonstrate that to you, I'm going to show you what cilia look like. So, okay, let me go to okay, I'm out of order here for whatever reason. Okay, sorry, forgive me. Okay, for some reason, it's not showing you how a normal cilia beat, but they beat very rapidly, about 10 cycles per second. These over here are cilia that you see in a condition called primary ciliary dyskinesia. These are individuals, because of a genetic defect in the cilia, they don't beat normally. They beat very stiffly. They look rather almost spastic. And you can easily imagine that this kind of motion is not particularly effective in getting mucus or bacteria to clear that from your lungs. Whereas, if you could see this, and if you come back, I'll show it to you, because we'll get this fixed for the next group, is that this beats very rapidly, very rhythmically, and you can easily imagine how this would sweep particulates from the lung. So in the process of taking care of children, we discovered a group of patients who seemed to be enriched with problems with cilia function, with this primary ciliary dyskinesia. And they're actually Amish. I was actually surprised. I grew up actually in an Amish community in northeastern Ohio. I'm not Amish myself, but I think it wasn't until I was about 10 before I realized you could actually buy cheese from someone who wasn't wearing a hat. But so I was surprised to see that there were as many Amish and Mennonite communities here in Missouri, and there was actually quite a few. And what's interesting about the Amish, as I think most everyone knows, is that they have certain rules that each of the communities have that include prohibitions and the limitation of electricity, telephone, automobiles. But what's fascinating is it's not the same in every community. Quite frankly, the community that we're talking about, their cell phones were better than mine. So they can adopt technology, but it has to be used primarily for work. Members rarely marry outside the church, and that's a very important part. So you have a lot of intermarriage that occurs within these very large communities. And these families keep extraordinary records, and they're also isolated from the mainstream. You don't find them, hanging around at the local schnooks very often. But because of these, they become very, very useful in genetic testing. So what we found was that in this single community in southwestern Missouri, and we've extended it beyond into Pennsylvania and to Wisconsin, that we're finding a large number of individuals from these Amish communities have primary ciliary dyskinesia. We've been able to identify a specific mutation that is pretty much unique to the Amish, this one right here. And we found this in Pennsylvania, we found this in Wisconsin, we have now found this in Arkansas in these Amish communities. So this appears to be a fairly pervasive mutation in this community. But what's fascinating in this is that we have also found a large number of patients with primary ciliary dyskinesia for whom we do not know what their mutation is. We do not know what the defect is. And again, by the support from the Children's Discovery Institute, we've been in the process of screening chromosomes and looking for this mutation. And we've been able to narrow it down into two relatively small regions. One small region on chromosome 12, another small region on chromosome 13, and we're in the process of doing sequencing so we can identify what the precise mutation is. And so we're going to really, I'm expecting in the next month or two, we're actually going to know what the mutations are. We're going to be able to fully genetically characterize these patients, and we're going to understand why they have primary ciliary dyskinesia. The question really gets down to what are we going to do about it? How are we going to treat this? And for that, I'm going to turn to Phil Bailey to tell you exactly how pond scum is helping us advance that purpose. So, not that Phil's pond scum, but.
All right. Well, we're off to a good start. I've been called an old guy and pond scum, so it can only get better. So. I'll turn my mic on and get on the record for now. So my job in this project, you may be wondering what a mechanical or aerospace engineer is doing working on this project. And there's several aims. One, which Tom really described, is to create the clinical and imaging and laboratory database that's involved in work with the Amish community. The second aim is to develop high-speed video microscopy methods to model and analyze cilia function. So when Tom has identified the genetic defect, we can reproduce this in a very interesting model and then describe quantitatively what's wrong with the cilia. And then there's a couple of other tasks which I will not go into. And this is such a privilege for a mechanical engineer to be able to contribute to a problem like this. It makes up for some of the other things I have to put up with, such as working with Tom. So this is the pond scum that we're talking about. It's actually a common algae called Chlamydomonas. And if you notice on the Chlamydomonas, there's these two structures that are called flagella, but they're actually wonderful replicas of the human cilia. And I'll let you read about Chlamydomonas, but what we really use it for is it provides a powerful model of pediatric disease. And it's a plant. You don't have to have human studies. You're not held up by the IRB. You can reproduce your genetic defect, not easily perhaps, but you can reproduce many of the things that go wrong with human cilia in a vegetative model. And we think we can use this model to actually test drugs that can restore ciliary function.